So one of the problems with chronic fatigue syndrome in the United States is that they don't like the name. So I think we should call it the Canadian name Carruthers syndrome, or the British name Ramsay syndrome. One of the things that I think that we sometimes forget about is what is the mechanism of the symptoms in chronic fatigue syndrome, ME. We've got fatigue, which is, supposed to be 50% disability. What is that? We have the others as well. I'm going to try to work a little bit of a way through this to try to build on what the speakers this morning have spoken about to try to help come to some resolution. I'm going to go very quickly through these first slides out of need for time and to emphasize the latter part. I came up with this little simple questionnaire where I used the Fukuda criteria and then had people grade their symptoms from 0 to 4. The 8 ancillary symptoms, I had them do a sum, the sum of 8 symptoms. And I came up with just a really brain-dead Mr. Moto kind of, master of the obvious kind of result. That on the right, you can see the people who have very little fatigue, and there were controls, and then we had people on this side of this red line who had a lot of fatigue. We drew a line across the red line here at 14 for the sum of 8 and said that the people who were in this lower right-hand corner were controls, the people in the upper left-hand corner were chronic fatigue, the people in the bottom were chronic idiopathic fatigue, and as Dr. Reeves predicted, we have CFS-like with insufficient fatigue syndrome, or out of respect to him, syphilis. One of the things that came out of this, though, is when we did a cluster analysis of the subjects. In our control group, we've got people that have no problems, people who have symptoms about trivial. Then in the control group, we have a group that have sleep as their biggest problem, and then sleep, headache, and myalgia. These are the control people. We often have talked about who our controls should be, and I think it's important to remember that they're not necessarily homogeneous. In the chronic fatigue subgroup, when we did cluster analysis, we have the group in reddish-purple here, in orange, it comes out, who have high scores for all of their symptoms. Close to them are another group that are in purple who have high scores for everything except sore throat and lymph nodes. Then we have another group that's high scores for fatigue, sleep, headache, cognition, exertional exhaustion, but then low for the last 4. And finally, a group that have somewhat mild symptoms throughout. From this, we came up with 4 clusters out of these 600 people. Now, these clusters will change depending upon the nature of the patients we recruit. But the point is, if we have distinct sets of symptoms and clusters, then we should have distinct combinations of pathophysiological mechanisms leading to these symptoms and potentially unique treatment plans. And much to my surprise, unique cerebrospinal fluid proteomes. Because these are our 4 clades of CFS people. You can see that what I've called C1 here has one particular pattern of proteins. What I've called C2 has a different pattern of proteins. C3, you can see they've got this hot spot, and C4 has this hot spot here. We'll be looking at these intensively to see if they do or can correlate with any brain regions, inflammation, or other central nervous system problems. So how many people have CFS, ME, and headaches? Only a few of you, I guess. What we've found is 75 to 80% of our people have migraines. We took the 2004 International Headache Association criteria, interviewed people during our 3 days, the 3 days they spend in hospital with us. We had many people develop their syndrome of usual headache, and were able to actually give them sumatriptan as a trial And I'd say that about a third of our patients went home with a new prescription and a new diagnosis of migraines. Two-thirds of the people had no aura. There's a thought that you have to have an aura in order to have a migraine. And in fact, that's very unusual. It's only in a third of people. If you have photophobia, phonophobia, nausea, throbbing unilateral headache, pain behind the eye, have to avoid usual activities, that is, lie down in a dark, quiet room, then you have a good chance of having migraines or a type of headache that's going to benefit from triptans or other migraine medications. And I think you should tell your GP that that's what you've got. You can go and take this paper of ours by Ravindran and ask him to at least give you a trial, take you seriously. I'll also bring up another point that 67% of migraine people have fibromyalgia. Are migraines a real disease? Is fibromyalgia a real disease? Is chronic fatigue syndrome a real disease? If there's a lot of overlap with a real disease like migraine, then do you think this one is? You think? This is another group. These are Gulf War illness patients. This is only 14, but you can see that in this 14, all but one had chronic fatigue syndrome criteria. Again, 80% had migraine diagnosis, again, mostly without aura. All of them met criteria for tension headaches. Sinus headaches, actually, they don't exist. Those people have non-allergic rhinitis, and it's the neurological non-allergic rhinitis of chronic fatigue syndrome. So I can give you the reference for that if you like. And then any headache, it was virtually the entire group. In the migraine community, the thought is that migraines result from central sensitization, which Dr. Davidson mentioned earlier. The thought is now, or one of the theories, that you have a intense neural depolarization in the cortex called cortical spreading depression. This is different from normal neural depolarization, where a single nerve would depolarize another neuron for a few hundred milliseconds. This can go on for perhaps a minute or longer. The cell runs a risk of dying during this period. Glutamate, potassium, hydrogen ions, MMP9, other proteins are released, and these can all initiate a depolarization of the adjacent cortex and stimulate the trigeminal nerves that would be in the leptomeningeal and dural areas above the cortex. These nerves, once stimulated, I should mention, with this cortical spreading depression, we're now looking for it in our patients. It is a spreading depression that moves outwards like ripples in a wave after you drop a rock. And using an fMRI technique, we think that we can now see these as these expanding concentric circles of depolarization. More on that later, hopefully. Now, when you depolarize these nerves, they have an axon response, which means that trigeminal neurons that innervate the vessels in the dura will now release CGRP and others that dilate these nerves, or these vessels. As they dilate, their walls stretch. The mechanical receptors in the walls will stretch, and they'll stimulate additional trigeminal neurons. And all of these will come angrily roaring through the trigeminal ganglion down into the trigeminal nucleus caudalis in the upper cervical spinal cord, where they have their primary synapse. And this primary synapse of incoming neural sensory neurons with the secondary interneuron is where the real central sensitization action is taking place. Get back to that later. This was mentioned by Dr. Davidson. Now, when you depolarize this ascending neuron, it goes to the thalamus. It also sends out branches to other regions within the reticular activating formation the Raphe nucleus, and you see both ascending and descending pathways activated that lead to an attempt to block this pain transmission signal here through the release of serotonin. That's why serotonin agonists are used for migraine. However, you also increase activity through the thalamus, which has connections with every other higher part of your brain. The hippocampus, prefrontal regions where you do your thinking, if you will, and your anterior cingulate cortex, which is right in here, that helps you decide what to do next. When you have a migraine, it's all screwed up because all of these areas may be firing at the same time in a discoordinated fashion, and you can't do anything. Sound familiar? Okay. There's a little easier theory. It's a nice new one. Doesn't have as many arrows. Basically, it says that within the brainstem, you have neurons that spontaneously or chaotically release nitric oxide and CGRP, or set up a network that does that, and it starts the entire migraine process from the brainstem, going up to the thalamus, up to the cortex, up to all of the executive functioning regions in the brain. Plus, it goes downwards to stimulate the trigeminal and induce the dilation that is, in this case, just a secondary incidental finding. Also, it will produce the lacrimation, the nasal stuffiness that can occur in some people with migraines. Notice the brainstem, because we're going to end up there. So, what about pain and tenderness? Does anybody have pain and tenderness? Myalgia, arthralgia, fancy names. This is sort of increased sensitivity to deep pressure. So, doesn't that mean fibromyalgia? So what are you doing here? So maybe they overlap? So why can't we get a rheumatologist here? I don't know. They don't talk to me either. As was mentioned earlier, the essence of central sensitization begins with an increase in sensitivity at the peripheral endings. You have proprioceptive endings that will be for joint motion, for innervation of the fascia over the muscle, and when these are stretched, you send a message to your brain. That's through highly myelinated fibers. They're not supposed to send pain messages. However, in the dorsal horn at the very back of your spinal cord, we have processes that go awry. These nerves come in and synapse, and then normally they would be gated out, as Dr. Davidson said. But we have other mechanisms that in chronic states, chronic inflammation, can lead to a loss of the antinociceptive systems. And she talked about the descending antinociceptive system with norepinephrine, serotonin, and dopamine. Mentioned the role of serotonin in migraine. The loss of norepinephrine release, particularly from the one area in the back of the brainstem called the locus ceruleus, can be very important. This is an area that when you need to focus, like right now, this area fires and stimulates what's called the dorsal attention areas in the brain. This network has alpha-1A adrenergic receptors, and you become focused on what you need to do. If it's a tiger that's standing in front of you, you're focused. You know what to do. However, if you overuse this system, you release too much norepinephrine, and now you start to stimulate beta receptors. Beta adrenergic receptors lead to cyclic AMP release, and it's in an uncoordinated fashion all over the cortex. What this causes is a dysfunction in your ability to organize your afferent sensory input. to your afferent interoceptive inputs from your gut, your bladder, other internal organs, the connections that you have to the amygdala, to your memory, to your sites of fear. You initiate your autonomic responses there, neuroendocrine, executive decision-making processes through the hypothalamus, through the anterior cingulate cortex. And one of the things that ends up happening is the brain doesn't act in a coordinated fashion to allow you to pay attention and focus. And instead, you slip into what's called the default mode pathway. This is where you kind of zone out. This is where you zone out. Wake up. Zoning out, the light is on, which means those specific regions of the brain now light up, but nobody's home. You're not responding. You're not concentrating. You can't do a task. If you were doing something, you just forgot what it was you were doing. Default mode intrusion is a mechanism we believe will play a role in this illness. And we hope that soon, when the light comes on, there will be somebody home. Looking at pain, we do dolorimetry, which means we take a spring-loaded gauge and we press it against people at the 18 traditional fibromyalgia tender points. We take the average of the kilograms of pressure, and we thought we would see an age-related decline in this measure or some kind of relationship. But here you see, if you're between 10 and 90, there's absolutely no relationship with age. This is unlike if you do manual tender point pressing with your thumb. However, if you bin each of these 1 kilogram numbers and count the type of people that are in them, you can develop a frequency plot where we look at the pressure threshold here against the distribution, the percentage in each group that would be at a particular threshold, like between 0 and 1 kilogram. The general trend here is that CFS people are over on this side. This is the spike for female CFS people. Male CFS people are here. Then we have normal females and healthy males. So the trend is male go to female. There's a genetic predisposition to this, I think. Males, females, you don't get it? CFS to healthy controls. I have a series of about 5 SNPs, gene polymorphisms, that I wish somebody would fund so that I can figure out what's controlling why different people have different thresholds for pain. Now let's get back to the central sensitization issue. As I mentioned with central sensitization, it starts in the periphery with an inflammatory process when studied in rodents, where you would inject mineral oil. I'm sorry, I won't do that to any of my people. But in allergic rhinitis, you do develop an increased sensitivity of the nerve endings. And that allows you to now respond to very low doses of pollen and start sneezing and stuffiness, but also respond to particulate and to bus pollution, exhaust particles, things that ordinarily you wouldn't respond to. The key step, as I mentioned, is the spinal sensitization that occurs in the dorsal horn, in the back of the spinal cord. And 2 key sensations that develop are hyperalgesia, which means that you would press and you now get pain, but with a lower pressure threshold. Or you can develop allodynia, or parallel pain, which means you may brush the side of your face with a feather, and now it gives you sharp, stabbing, burning electrical pain, as in trigeminal neuralgia. I believe that we will show that similar mechanisms that have been identified in the spinal cord will also take place in higher centers in the above the spinal cord, in the thalamus and cortex. I'm going to flip over these, which Dr. Davidson mentioned, and instead focus on the postsynaptic neuron. And this is the model that comes mostly from rodents in the spinal cord. You'd have the nerve coming from the paw, for instance, and then you would have this nerve that carries the signal of pain up to the brain. You can see there's a whole bunch of receptors here that can respond. But the critical one is to have an increase in the amount of glutamate that is released and have it act on what's called the AMPA receptor. In this case, this AMPA receptor is of a type that is very, very permeable to calcium. So lots of calcium floods in, it stimulates more calcium to flood in, and that leads to depolarization of this nerve and transmission of the pain message. In addition, that calcium also activates phosphokinase C, and probably phosphokinase C eta and potentially zeta, or other as yet unknown isoforms that I believe will turn out to be critical new targets for drug therapy. There are others that are also activated in this process, but I think this fundamental glutamate, AMPA, calcium, PKC, and downstream transcription factor pathway is ideal for us to be looking for new drugs. You can see the central nature of PKC because everything activates it. A key player in this upregulation of these neurons, as was mentioned earlier, is the microglial cell. And it turns out, sadly, that nerves are their own worst enemies in a way, because when a nerve gets injured, it'll release a protein, mu-calpain, which is a cysteine protease. The cysteine protease then stimulates NADPH oxidase that releases superoxide anions, which you may know better as bleach. When the bleach diffuses out, what does it do to the neuron? Starts to kill it, neuron gets hurt, releases more mu-calpain, that releases more superoxide anion, and you've got a vicious circle here. Over the course of months to years, you can kill off these nerves, make the microglia angry, and you'll end up with what has been termed reactive microgliosis. We need brains, brainstems, in order to see if this is true. I'm not advocating that anybody go out and die quickly. If you look at this process, this interaction between the postsynaptic neuron here and the microglia cell, you can see that it's a little bit more complicated. But that complication adds some new wrinkles to what we can look for new therapies. Again, here with the AMPA receptor, we have this glutamate 1 receptor that should be a very good target for us. We have PKC activation. We have fractalkine and MCP1 that are released by injured neurons or activated neurons that activate the microglia. We have the mu-calpain that could be antagonized. And if we have antioxidants, we might be able to decrease the reciprocal damage here. Earlier, we talked about ATP that's released, potassium ion, and hydrogen ions that are released during injury. It also turns out that with neuron injury, you release a miRNA called LET7. It's part of a small family of these microRNAs that act on toll-like receptor 4 to produce a nerve injury. And it turns out neurons themselves can release this miRNA that acts on TLR4 that leads to further toxicity of adjacent neurons. That would be a good target. Another one is Reservatrol, which is the colored agent in red wine. but I suggest you stick to grape juice or tablets and stay away from the alcohol. Tricyclic antidepressants, if we had more selective ones, because these probably work through PKC-mediated mechanisms. Antioxidants. And then one that I think is growing in appeal is to begin to use endogenous endocannabinoids. Marijuana has many different lipids in it, resins, in addition to the delta-9 THC, which interacts on its cannabinoid receptor, but also on the capsaicin or chili pepper receptor. We have about 60 other complex fats. And I believe some of those will turn out to be extremely useful for dampening down the overall inflammation, neuroinflammation that we see. The odd thing about endocannabinoids is that when a postsynaptic nerve is excited, you metabolize the membrane fats or lipids, produce the endocannabinoids that then go back to the presynaptic neuron and can inhibit. And an agonist may also be able to diffuse in and inhibit the synapse. From what I'm told, cookies are better than smoking it because of the lung injury, but also better absorption of these odd other lipids. I take that from my patients because my patients teach me lots of things. Does anybody have brain fog now? Oh, damn it. I thought I'd keep your attention. This is the study by Barden and company, which I think is fundamentally important. It is a very, very, very difficult paper to read. It's a very difficult paper for me to talk about. So all I'm going to do is show you the pictures. And all you have to read are the things that are in yellow. They looked at the amount of gray matter that was in the cortex or nuclear regions within the brainstem. They also found in this particular group that they had a problem with an increase in diastolic blood pressure. And this reduced their difference between systolic and diastolic pressure. This graph is, or this picture is what happens when you multiply the group, CFS versus controls, times the pulse pressure, and then try to correlate it with the thickness of the gray matter, adjusted for, never mind. Anyway, this is a measure of how much gray matter there is and the decrease that you have in chronic fatigue syndrome compared to controls. This is your brainstem. This is where all the nerves come up from your spinal cord and all your nerves go down through your spinal cord, to your spinal cord. This is the reptilian brain that keeps us all alive. In particular, you have this area, the reticular activating substance that's right in the center, the periaqueductal gray matter. This is vital for life. This is like your body's alarm clock. Not that it wakes you up from sleep, but when that tiger jumps out from you, this is the area that responds. And this is the area that seems to be most affected in this particular study. All of the nuclei that are present in the brainstem here regulate things like sleep, the transmission of pain. There are many fine-tuning motor and other sensory nuclei here. The significance of the differences is very impressive, and I think this is where we're going to find the ultimate lesions in ME/CFS. As far as the white matter is concerned, this is the axons. We have, again, damage in a very similar area. In this case, the corticospinal tract that carries nerves down to the muscle neurons, the hypothalamus, which regulates all of your hormones. And the reason I blew this picture up is so that you could see that here they found this incredibly tight correlation between the loss of white matter and the duration of fatigue. And they predicted shrinkage at about 1% per year. As I agree with the authors, this needs to be replicated in an independent group of people. But defects here could lead to most of the symptoms and problems we've discussed. Sleep? Anybody have sleep problems? Well, brainstem's involved. I'm going to close with some of our own work. We took people and did an fMRI with a working memory test. Then we had them exercise. The next day, we had them exercise and then repeated the fMRI. You can see here are healthy controls. And this was in Gulf War, plus people who had Gulf War who also met CFS criteria. You can see our controls have a very high accuracy on both days. Our Gulf War CFS group didn't. They had a tremendous variation. But what was most striking was that as a result of exercise, these people had an increase in their accuracy. They're the increasers. And we had some people who dropped off a little bit. There are decreasers. They kind of went to hell in a handbasket. This is a heterogeneous condition. What's the difference between the two? fMRI, this is your brain if somebody cut it off right through here. This is a control area. This is a control picture before exercise while doing this working memory task. You can see this area is part of the dorsal attention network that you use for concentration. When you want to remember a short string of numbers or letters, A, B, C, D, you activate this working memory region in the inferior parietal lobe back here. And then when you want to push a button to report back that you recognize and remember a letter that you've seen, you push this supplementary, or you activate this supplementary motor area. So this is a normal response. You can see that after exercise, the healthies, the controls, hardly light anything up. They have a little bit as far as fingering, but they've developed what's called automaticity. That is, they've learned. In the increaser group, they don't activate their dorsal attention network on day one, and this may account for why they don't do as well on the first day. They do activate their working memory regions and their supplementary motor area. However, on day 2, you see nothing in the attention areas. And they, unlike the controls, still have to use this working memory region. So in order for them to have an improvement in their score, they have to work really hard, and it's tiring. So the increasers still need their inferior parietal region. What about the decreasers? Well, here's our controls, our increasers. Do you see anything different in the decreasers? You can see that here they've activated their dorsal attention network, and you can see that they've had to recruit almost 4 times as much the volume in their working memory in order to remember what order the 4 letters came. So they are working really hard, using up tremendous brain reserve, cognitive reserve, before exercise. After exercise, they don't activate anything. They can't activate their areas in the dorsal attention network that are necessary for concentrating, for focus. They can't remember the letters, and therefore they can't stimulate their fingers to do anything. Again, this is only one section through the brain, but this is a series of sections that show clearly the distinction between these phenotypes. Now, I'll close now. I think that fatigue we will redefine as being more an emotion and more a consequence of all of the other symptoms that Fukuda and company selected. As I showed, migraines are probably the cause of most of the headaches and are associated with central sensitization. The myalgia, arthralgia, sore throats, sore lymph nodes are central sensitization with hyperalgesia, with interoceptive dysfunction, and the neurocognitive changes, the severe fatigue with exertion and sleep disturbances, probably come from the fundamental defect in brainstem and other central nervous system alterations. I'd like to thank you for the invitation and the opportunity to present this for the first time ever. Thank you.